Hey, welcome everybody to microorganisms, or microbiomes actually, 1.3. Um, so today we're going to learn more about microorganisms and talk about more about how small they are, and talk about the size of actually molecules too. So first things first, let's jump to our slides. And what is the first thing they're going to have us do? The warm up. Consistency. I like that. I like it when you get into a routine. So you're going to go to this Amplify website. Looks a little bit different than this, obviously. It looks more like, boom, this, and you're going to answer this warm-up. You've been learning about tiny organisms that are made of a single cell, but are there objects even smaller than a cell? Sounds kind of like a leading question to me. It says, choose the statement below that you agree with. You can choose as many as you want. It's okay if you aren't sure. Oh, geez, that takes the pressure off, doesn't it? So choose which of these that you agree with and then explain why. Why did you choose these statements? I gotta answer why? Yeah, you gotta answer why. You gotta explain it. Don't just pick random things, but tell me why you picked random things. Anyway, so go ahead, actually, first thing, hit pause, do that right now. Come back to me when you're done. And we are back. So now we're going to actually jump to activity 2, comparing the scale of cells and molecules. And really, I'm going to just talk at you for a little bit. Let's go through these slides. Hey, it's Dr. Mara. I remember Dr. Mara. She had a job for us. So remember that you are learning about the scale of microscopic things as part of your role as student researchers for the Microbiome Research Institute. That's right. We had to find out about these things. Okay, yesterday, we were asked to draw scale models. So we were supposed to pick a tiny organism, one of those microscopic organisms, and we were supposed to draw it 20,000 times bigger than it actually is. So when it gave us the measurement in micrometers, we were supposed to multiply that times 2 centimeters, 1 micron, and then we were supposed to draw that many centimeters long. So for instance, if your organism was 2 micrometers long, you were going to draw it as if it were 4 centimeters big. Okay. Now, keeping that in mind, those are 20,000 times bigger. We talked about molecules in our warm-up. That was some of the statements that we had. So what are molecules? What do you already know about them? Think about that for a second. Just go and take some time. Think about that. What are molecules? What do you already know about them? I really don't know much about them, Mr. Reagan. Can you tell me more? Yeah, sure. Let's talk more about them. Molecules, when we did our card sort, we had a water molecule, didn't we? And look at this. People often get confused about the difference between cells and molecules. Which are bigger? Are cells bigger than molecules? Are molecules bigger than cells? Are they like the same thing? All living things are made of cells. Okay, so where do molecules fit in? We know the size of cells. So if we knew the size of a molecule, we could compare them. All right, great. So cells, remember we drew them 20,000 times bigger, right? We measure cells in micrometers, okay. But what about this, nanometers? Well, let's talk about the size of a nanometer. So if a micrometer is 1/1000th of a millimeter, a nanometer is 1/1000th of a micrometer. That's a lot smaller. That is way, way smaller. So, okay, think of a millimeter in terms of like a meter, okay? Now, how much smaller is that than a meter? Now, think of a micrometer, that's the same difference as a millimeter and a meter, but, to a millimeter. And now a nanometer is 1,000 times smaller than that. We are getting down to a very, very, very small scale here. So the width of a DNA strand, have you heard of DNA before? Deoxyribonucleic acid. Something that we might measure in nanometers. We're talking about something on more of a molecular scale here. How would we represent a molecule on our scale model drawing? We drew our organisms, but how would we show a molecule? How big do you think it would be? Let's do this. We're going to try to represent it on our scale model drawing. So on one of the microorganisms, draw the smallest dot that you can. So you drew an organism that was 20,000 times bigger than it actually is. Now draw a tiny, tiny, tiny dot on one of those organisms. How many molecules do you think fit in that dot? One molecule? And again, we're talking about 20,000 times bigger. So do you think one molecule might fit in there? 10 molecules? 100 molecules? No, you're all wrong. 200,000 molecules. You drew that 20,000 times bigger microorganism. And then you put a tiny, tiny dot on it, the smallest dot you could make. And in that tiny dot, still 200,000 molecules would fit in that tiny dot. So microorganisms, yeah, they're really small. But guess what's even way, way, way smaller? Molecules. Molecules make up those microorganisms. So just like, kind of like how you and I were made out of tiny little cells that are microscopic, you can't see them. Cells are also made of molecules. They serve different purposes and different functions, but on a scale size, you can sort of think of it that way. We're going to jump to activity 3. Let's go there. Mr. Wiggin, did I have to answer anything here? No, you didn't. Student-to-student discussion. Great. What is that going to be like? So each of these tiny organisms is just one cell. We took a look at, what was that C? It's hard to read. C. difficile bacteria, and then the ringworm fungus. They're microscopic. They are one cell big. You would not be able to see them with the naked eye. Let's think about our unit question. So if we have 100 trillion of these microorganisms living on us and in us, how could that help keep the body healthy? We're going to keep coming back to that question all unit, the entire unit. So here's a good question here. Can you observe microorganisms on your hand? Let's make sure we find out. Let's see. Look. Can you observe microorganisms on your hand? You're going to actually answer in activity 3 on Amplify. You're going to just choose yes or no. Look at your hand. Can you? Do you think there are any microorganisms on your hand? Well, if you've been paying attention, you'll probably say, yeah, I think there are. I'm not really quite sure what they are. All right. So what are we going to do next? We're going to talk about how could we see some of those microorganisms. And here's how we would do it. Sometimes we can see them, even without a microscope. And the reason we can is because there are so many of them. So think about this. Imagine you're looking across the street, and there's a grain of sand across the street, right? Would you be able to see that tiny grain of sand? No, it's like all the way across the street. Like it's so small. But what if there was a whole pile of sand across the street? Would you be able to see that? Oh, okay. Yeah, I might be able to see that pile of sand. Well, in that pile of sand across the street, would you be able to still see those individual grains if you were on the other side? Probably not. I'd probably be able to see the pile, but I couldn't see the individual grains. Well, how could you see the grains? I don't know. Get a binoculars or a telescope or something. So one way that scientists observe microorganisms is by letting them multiply. So it's not just one organism, but it's a whole pile of microorganisms. And that's called growing a culture. So let's talk about what that means and how they might do that. So let's say you wanted to study the microorganisms on your hand. They would take a swab. Oh, we all know what swabs are now, right? Very familiar with that. And you would rub it across your hand. And the idea is that some of the microorganisms on there are going to stick to the swab, okay? Then what do? You take a petri dish with this, basically it's a food for the microorganisms that's inside of it, and you rub it on there. So some of the microorganisms are going to rub off of your swab into this petri dish, into this big thing of food. They are happy. If some of them fall off over time, you're going to see some grow. You got to give it time. You got to let them grow. And it's not recommended that you try this at home because sometimes, you might grow some bad microorganisms. Here we go. Yeah, we are not going to grow our own. Number one, it takes a while. And number two, it's just not safe to grow our own microorganisms in that type of environment, in a non-lab environment. So let's imagine that we did. Take a look at this. And actually, let's make sure, where are we at here? Let's go to next. Here we go. Right here. This photo shows the petri dish on day 1 of the investigation. Discuss and record your observations and predict what you'll observe on day 5. So what do you see here? I see nothing. I I don't see anything. Maybe you do. Maybe your eyes are better than me. So can you see evidence of microorganisms? I would say, no, I can't. But what do you think it's going to look like on day 5? What do you see in this Petri dish? Take a second, write down those observations and those answers. Go ahead and take a minute, do that right now, and then come back to me. Uh-oh, the slides are wrong. Amplify, better fix that. So instead of showing us the Petri dish on day 5, they actually just repeated the photo from day 1. Let's actually go to the lesson, though, because they did it right. Let's go to number 3. Activity 3, part 3. Now this is what it should look like on day 5. We're going to answer these same questions. Do you see evidence of microorganisms? Well, let me give you a hint. These little dots here, those are colonies of microorganisms. Make a prediction. What do you think it's going to look like on day 9? Describe what you observe in the Petri dish. What do you see here? Okay, take a minute, do that right now. That was my favorite part. You wrote down all your observations and it said, hey, what do you think it's going to look like on day 9? Do we get to see it on day 9? No, we're going to do this other thing next. Hey, but don't worry. I promise we will look at it on day 9, just not today. So we're going to do this. It's called word relationships. And this is something we're going to do multiple times throughout the year. Going back to our chapter 1 question, how small are the microorganisms that live on and in the human body? We've been talking about this. This has been all of chapter 1. We're trying to figure out, wrap our minds around how big are these things. We went over some of these words: cell, microorganism, nanometer, microscopic, micrometer, and molecule. What we're going to try to do is we're going to try to see, can we use these words intelligently, like we know what we're talking about? And we will use word cards, I don't have word cards, to create sentences that answer the chapter 1 question. This question: how small are those microorganisms? Okay. Now, what they'll expect you to do is work with a partner. Hey, I don't know, go find someone in the class and chat with them. Send them an email. Try to connect somehow and talk about these words. Help each other out. The way that it works is you want to use as many of the words as you can. You want to use at least 2 words in every sentence that you do. Come up with multiple sentences. Try to get as detailed as you can. Try to, come up with something that actually sounds pretty intelligent. And then, where do we write those words? Well, you don't. You don't. Oh, so you mean I don't have to do it, Mr. Wiggin? No, don't sell yourself short. Try. Try to do it. Because this is good practice. Because if you can do this, and if you go through the work of like really trying to understand these words, that is going to help you out immensely when it comes to the test. When it comes to you having to show me what you know. Okay? So make sure you do that. Try that out. Try to answer that chapter 1 question using these words. All right, let's wrap that part up. You've learned a lot about the size of microorganisms since you wrote a response to our chapter 1 question in the very first lesson of this unit. You will write a new response to the question so you can more fully explain what you have learned. Where do I do that? Well, not here. You're going to do that in the homework right here. So I think that's like one of our last slides. Yeah, the homework. And basically, what do you know? It's just like what you just did with the word relationship routine. You're going to try to use these words, and you're going to try to answer that chapter 1 question: how small are the microorganisms that live on and in the human body? So remember how you said, oh, okay, so I just don't have to do it? Well, you do. You have to write it out right here. So make sure you do the homework, answer that question. Try to use as many of these words as possible in your response, and make sure you hit hand in, and I will see you next time.